Okay. Am I good to go?
Yes.
Okay. Good afternoon, everybody. My name's Mebs. I'm the Managing Director for Swift Analytical. On behalf of my colleagues at Swift Analytical and Fluicell, I'd like to welcome you all to today's webinar. We really appreciate you giving up your time today to join us, and we hope you continue to stay safe and well during these difficult times. Just to let you know, before we begin, all microphones and cameras, apart from those of the presenters, will be switched off. You will have a chance to ask any questions. Please add them into the Q&A section at the bottom of the page, and we'll be happy to answer these after the talk. So, a little bit about Swift Analytical. Swift Analytical is based in the historic medieval city of York. If anyone has ever visited the city, you'll know that it's famous for a number of things, including the Minster, the city walls, its chocolate, and more recently, it's become home to wizards and elves as muggles flock to visit the shambles, which people say resembles Diagon Alley from the Harry Potter stories. We will be celebrating our 10-year anniversary in February 2021, and we'd like to take the opportunity to thank our employees, our customers, our suppliers for helping us with this journey. Our focus is to introduce exciting new technologies to researchers in cell culture, cell technologies, tissue engineering, 3D bioprinting to enable assay development for drug discovery. So on the next slide, we'll see a quick overview of some of the related products to this community. So if you go to at the top right, you'll see the Biofluidics BioSpot system. And this is a non-contact nanoliter-based liquid handling system. And it can be used for a variety of applications, but for this community, it can be used for rapid dispensing of cells whilst maintaining high cell viability. So for example, a 1536 well plate can be filled with cells in under 20 seconds. At the bottom of the page, you will see the cell dynamics weight system. And this is a unique flow-based device which simultaneously measures the weight, size, and density of spheroid-like materials faster and more efficiently than existing systems. On the top left, what we are doing is we are helping to reduce the amount of plastic weight from cell counting workflows by use of the Imrali Inventions iWash system. The iWash system can wash and recycle plastic slides in less than 20 seconds and can allow the reuse of plastic slides for up to 50 times. This not only reduces plastic waste, but also impacts on laboratory running costs. So on the next slide, we are proud to be the UK distributor for the Fluicell. So on the top right, you will see the BioPen. The BioPen is a platform technology which is being used in a number of applications around the world. And these will be discussed in more detail. At the bottom of the page, you will see the Biopixlar, which will be the focus of today's talk. So that's my introduction. What I'd like to do now is to hand the presentation over to Matija. Matija has a PhD in molecular and cell biology and has worked with life science instrumentation companies for the past 7 years. Matija is the Chief Sales Officer for Fluicell. So thank you, Matija, over to you.
Thank you, Maps, for this kind of introduction and also for organizing this event with us. And in my behalf, welcome to all of you here. So for today's talk, I want to present our company, our technology, and some of the applications that are coming from the technology. So Fluicell is a company based in Gothenburg, Sweden, And we are really a provider of microfluidic solutions for life sciences. Our technology is based upon controlling the flow of liquids in very small volumes, enabling access to individual cell biology and its microenvironment, and better utilization of high-value samples. So for the talk today, I really want to focus more on the technology side and the application side, and not so much on the commercial side of this. So if you have any questions on the commercial part, please don't hesitate to contact us after the event. But as a general overview, this is our platform family for single cell investigations. We work in two main areas of single cell investigations, one being compound or solution delivery to single cells. And in this area, we have two platforms, one being Dynaflow Resolve, which is an ion channel screening platform for single cell patch clamp recordings, and another BioPen, which is a localized delivery system for targeting single cells inside an in vitro cultures. The other part of our technology is the bioprinting. So here on the right hand side, you see Biopixlar, which is a 3D single cell bioprinting platform. For today, I will focus mostly on the Biopixlar in the technology, and at the end, I have a couple of slides showing also BioPen. BioPen and Biopixlar share the core technology, but of course applied in a bit of a different area. So starting with bioprinting. Bioprinting is an additive manufacturing process using biological materials. There are many technologies in bioprinting, and many of those have actually been developed using existing printing technologies, but were optimized for handling biological materials. In bioprinting, it's very common to use cells printed in a binding material, kind of biogel matrix, that is used for positioning and for tissue structure formation. Our bioprinting platform is called Biopixlar, and it has a very unique technology to what I've described before, and also a unique application areas. So our technology is really focused on 3D single cell bioprinting, and as a first main difference, it's the printing process does not involve any kind of binding matrices. So we really print cells directly in cell suspensions or their growth media. So our approach here in a couple of points, and this goes back to our core technology. Our core technology is microfluidics. So you see an example of the microfluidics printhead here on the right hand side. And in our approach, the cells are housed in this microfluidic printhead in the containers, and then pushed with an air pressure through the microfluidics channels inside the print area, directly in their growth media, and then being printed on a substrate in the print area. Our focus is really on single cell precision of printing to enable high resolution biology, rather than printing kind of big organs. So now going from the printhead to the system, the microfluidics printhead you see here is integrated into the a holder that is served for positioning of the printhead inside the cell culture environment, and is also applying kind of the air pressure to the cell suspensions inside the printhead. Here is an animation of the approach that we are using. So here is the printhead now, obviously without the holder around it for better illustrations. We see different containers that can house different cell types. So for example, in this case, we have a cell type 1 suspension in this container. We apply air pressure to this particular container, and the suspension is pushed through the microfluidics channels inside the print area. it's very easy to switch the air pressure from one cell container to another cell container. And this cell container can house a different cell type suspension. So what this means is that you can on the fly switch the air pressure to a different container, and now the other container suspension will be pushed inside the environment, resulting in kind of creating multicellular structures basically on the fly. Now, coming here to the right-hand side is how this works the way it works. This goes back to our design of the fluidics. At the end of the print head, which is looking into the printing area, you have 3 channels. Through the middle channel, the cells are pushed inside the environment in the print area. And the channels next to it are basically vacuum channels. So what happens is this creates a recirculation flow where cells are coming out and are then immediately vacuumed back into the system. So now we enter another aspect of our approach. In this recirculation flow outside of the system, we combine this with the cell adhesion pre-coating or substrate. So the cells come out, they either attach to the surface, or if they don't attach to the surface, they go back into the system. And this leaves no free-floating cells around in the cell culture, and really enables very high precision of the printing. Here is kind of the video how this looks in live. As you see, our focus here is really the precision of placing the cells in the very defined area. Just for reference, the square here is 500 times 500 microns. So this is the basic of how we print cells. Now the next step is how we come to printing a 3D structure. Here we rely on the very same approach described before. So combining our fluidics flow with adhesion layers. So for 3D approach, we first position a cell adhesion layer on top of the existing confluent structure of cells, and then we position the next layer on top of that. We can repeat this process and have, in this case, 3 cell layers. I want to mention that this cell adhesion layer, this is really not kind of a physical barrier between the cells, but it's rather kind of a molecular layer of cell adhesion molecules with the goal to attract the cells during the microfluidics flow to stay in the area where they are positioned. And through time, the cells between the layers then start to forming a 3D structure. In this example, you see A431 skin cancer cells on the bottom marked green. We have a HACAT epithelial cells in the middle, and another A431 cell layer on top of it, and a micrograph representation of this. Here we have a video of how this looks in real time. In this example, we have a fibroblast layer in the first layer. We print the HepG2 cells, so the hepatocytes, in the second layer. You can also see very nicely in this example how the cell flow is going on. So the cells are coming out, some of them are attaching to the surface, and if not, they are immediately aspirated back into the system. And at the end, you saw the cut of the flow. So that's a thing that we control. The flow can be very well controlled. In this example, we also printed fibroblasts in the second layer next to the hepatocyte patch, and I will come to this a bit later. Another aspect of our technologies is the flexibility. So as you see in this example, we can also print very customized shapes. The reason for that is that the system has an integrated imaging into the workflow and has a kind of joystick control. So you can visually create customized shapes or tissues. It's very well suited, for example, for kind of creating very customized microenvironment or something similar. Now going to some of the capabilities and examples. As I mentioned, the core of our process is that we control the flow of the speed. We control the time of the flow, so the flow can be cut at any point in time. And also, it's very flexible to control the density of the original cell suspension. So with all those variables can be suited to particular application. We can position single cells very precisely in the cell culture media, as shown here, expand to kind of clusters of cells of different ratios of cells, to 2D models with different cell types, so multicellular 2D models, as well as expanding into a 3D models, as shown here. Another point to highlight is the formation of tissues, as for example seen in this 2D model. Because we can position the cells very precisely and also very close together, we can get very good results very fast. So for example, in this case, we have a print, an image immediately after print and a confluency forming after 24 hours. And this has been generated with very limited cell counts. Here, at this point, I want to highlight a paper by Jeffries et al. In Scientific Reports, which was published actually last week. And in this paper, it's really all of the concepts that I have described today and are presented. So I would really invite you to look at this paper up and go into more details about these concepts and technology. But from the paper, I also want to take a couple of examples. So skin cancer cells with HayCat epithelial layer around them, print at 0 hours and 20 hours in different shapes and in different cell ratios. We also have kind of a basic skin model with epithelial HayCat cells, either differentiated or non-differentiated, where we have used the Seiki 10 marker, here shown as green for showing the differentiation. And in this experiment, we wanted to see the effect of retinoic acid on this model, the retinoic acid being a known proliferator of epithelial cells. And another very important point with our technology is basically what we do with printing, meaning that we print in cell culture media, in normal growth media of cells. We also have optimized the flows and stress levels of the process on the cells. Our technology is very well suited for printing sensitive cell types. So in this case here we have an acutely isolated dorsal root ganglion neurons from rats, 9 days after printing, showing a very nice formation of inter-cell networks. Another application I want to show here is kind of bioengineering of liver model. So what we try to do here is show the different examples of how we can do a liver model. So we had a monoculture tissue, which means that we just printed hepatocytes in a very specific area. We did a 2D tissue, meaning that we printed hepatocytes, surrounded them with fibroblasts, so a 2 culture tissue. And we did a 3D tissue, meaning we used a fibroblast as a first layer, and then on top of the fibroblast, we first printed a patch of hepatocytes and surround them with fibroblasts. So this is the example that was shown in the video before. Further on, we wanted to test functionality of this. So we used albumin as a marker for liver metabolic activity. and basically monitored albumin secretion in these tissues. And what we have found out is that the 3D tissue has a significantly higher albumin secretion after 7 days compared to 2D tissue and the monoculture tissue. This is especially interested between these two results because you see a big difference in 2D and 3D tissue, and the difference is really the layer of fibroblasts beneath the 3D tissue. The last couple of slides on bioprinting, I have taken them almost directly from our R&D, so these are not published, but I think they are important to show the points from what I mentioned before. Our technology is very well suited for printing sensitive cell types, like for example stem cells. So again, in this example, we focused on printing stem cells in a very defined area of this square. So these are the iPSCs directly after print, and iPSCs after 24 hours in the bright field, and also a fluorescent image here. And the last example at the end are the mesenchymal stem cells, which are marked here in yellow. And these were printed on top of a fibroblast layer in green. So fibroblast is the layer 1, and the MSCs were printed on top of them in the second layer. And again, in a very defined area. And some further work on the neurons. We worked with neurons with our partners from Selectricon. This is a company here close to us in Gothenburg, who is specialized in neuronal applications. What we did here is printed cortical neurons acutely isolated from rats. Again, the point was to really print these neurons closely together in a defined area and see the development of these prints. We see after 5 days that our prints start to develop some interconnections. And as a reference, this is a control sample where cells were, these primary neurons were just normally seeded in the cell dish. So obviously, with normal seeding, the cells are further spread around, so the connections do not yet develop so fast. They do after a couple of days later, and that's what kind of we wanted to see. And further follow up on the prints, neuron printing. So we also did nicer images after 14 days of incubation to really check about these formed interconnected networks between the neurons. So I have this for the Biopixlar. I hope I have shown really that the focus for us is high precision and printing of multicellular models with high resolution to really kind of have a customized shapes of these prints, for example, in kind of modeling microenvironment, but also to show the printing of sensitive cells with high viability. Since, as I mentioned, compared to other bioprinting technologies, we really print on directly in growth media and kind of excluding some of the factors that can lead to low viability in the bioprinting. And for the last 5 minutes, I also want to show more about our compound delivery platform, BioPen. As I also see this as a very exciting technology in the field. Actually, evolution-wise, we first started with BioPen. So the BioPen was the first platform that we had. And then it was evolution, we went from compound delivery idea through this technology to actual delivery of cell suspension or cell printing or bioprinting. But BioPen, it's a platform that directly targets individual cells or small group of cells in vitro. You have full control of position and timing of the delivery. And with the BioPen, you kind of create a microenvironment around the cell and without any dissolution into the surrounding areas. And this is exactly what is shown here. So you see the, you see the concept being exactly the same as in the bioprinting. You have 3 channels looking into the cell dish. For the middle one, the solution is pushed into the area, and the channels next to it are vacuum channels. So the solution kind of comes out and is then vacuumed back into the system. And this creates a microenvironment from where there is no dissolution in the surrounding area. And you can use this to position it next to a cell, single cell in vitro culture, and create a microenvironment around this. With BioPen, you can create a microenvironment for a continuous flow for about an hour or so, or you can also deliver very fast pulses. For example, a second pulse, a 1 second pulse can be delivered to a cell. Again, we control the flow of the speed as well as the time of the speed. So this is why this is enabled. And now going into a couple of applications here, really using BioPen for single cell studies, a paper from 2 years ago. They use BioPen to actually study the retraction of a cell protrusion. So what happened is that they induced the cell protrusion, the retraction of the cell protrusion with BioPen by delivering a PDGF to a cell protrusion, as shown here in a timeline perspective. So delivering PDGF with BioPen on the cell protrusion, and then seeing how the protrusion is retracted. And in the meantime, following the phosphorylation on that retraction of the protrusion. Another example is in cell networks, so cell-to-cell molecular transport, a paper from 2015. They used BioPen to deliver a menthol solution in combination with a calcium infused buffer to a cell in the network, and then study the calcium transport through tubular interconnections. So in the network, they targeted a single cell, which was here showing the affected cell by the BioPen. However, the cell next to it, which was connected with the network, was not affected because, as we said, the flow of the BioPen enables a very local targeting or very local delivery of solutions, and there is no dissolution into the surrounding area. The same concept can also be applied, for example, in neuronal networks, targeting a single neuron in a network and trying to see network-wide response to certain compounds. And the last application, which is also the latest paper from this year, from Waschal Lab at the Rockefeller University, is studying, they study mosquitoes, and they have used BioPen in this example to deliver whole blood to a tip of a mosquito blood feeding needle. So the idea was to observe neuronal responses from mosquito to whole blood, and they needed something to very precisely deliver whole blood to the tip of the mosquito. So this was done using the BioPen. And with this, I would like to thank you all for joining here. And as Maps mentioned, please use the Q&A function in the platform to write your questions. And you also have our contact details here. So for myself and for Maps, who is our partner in the UK. Thank you.
Thank you for that, Matija. Really great overview of the BioPen technology. We do have quite a few questions, so if you don't mind, we'll spend the next few minutes asking you the questions on behalf of a number of the attendees. So the first question is from Artemis Stamboulis from the University of Birmingham, and she'd like to know is, what about the shear stresses inside of the container? Do they affect the integrity of the cells?
The stress throughout the system, so also the container and through the flow, are optimized for basically cell integrity and cell viability. So it's as I mentioned, the whole system is optimized in this way because we know that's a very important part of the process.
Okay, thank you. So the next question is, does it print in certain patterns? And yes, I think we've demonstrated from your presentation that it does. We have total control of the printing capability. Would you like to add anything else to that?
No, I think it's fair. Yes, it can print patterns, and basically how it works is that we have a XY stage beneath the print head, and you have full control of the XY positioning, right? So that's how you can create patterns. Yes.
Okay. So the next question is from Sajad Nampur. And the question says, is it possible to use soft hydrogels for printing in this system?
So this is a question we get quite a lot. We we started, or the focus for us is printing directly from cell suspensions. Viscosity is a limitation for the microfluidic systems that we have. And on the other hand, another important feature is the resolution, right? So the accuracy of placing the cells, basically one cell next to each other, is based on the fact that you remove the fluid, that you print in the cell culture media, basically. About the soft gels and what type of gels, it's really a question of that we would need to spend in more details, discussing what is the exact application and what is the expected result of that, and also to see what is the viscosity of that. So it's really hard to say in general yes or no.
Okay, thank you for that. So the next question is to do with the binding layer of the 3D printed cells. Are you using a protein such as fibronectin?
The binding layer, it's exactly. So the concept of the binding layer is really cell adhesion of cells. So all any molecular, being a proteins, being a polypeptides, that are used in those cell adhesion mixes. That's the idea behind that, right? Again, what is the best possible protein polypeptide for that mix depends a bit on the cell type. But yes, also proteins, I think, like have fibronectin, which are known to be involved in cell adhesion, is something that would probably be a good fit for that. Yes.
Okay, thank you. So the next question is about the speed of the printing process. How much time does it take for cells to stick to the surface?
This is a continuous process. So as we saw in the videos, it's a continuous flow. The cells continue to flow, and there is a certain percentage of the cells that basically stick to the process. What we control is the flow of the speed. So if you want to have a higher printing process, you can increase the flow of the cells, increase the density of the cells. What you're losing with that is, again, the precision, right? If you want to really have really precise single small cells, then you have to be a bit more patient. If it's really about bulk printing with limitations, obviously our technology, as I think I mentioned on the second slide, it's not it's not expected to print multiple decimeter type tissues just like that, right? But if you want to print with higher speed or higher cell counts, that's also possible, right? So it's always a question what is the expected result and what is the what are the cell types and so on.
Okay, great, thank you. So the next question is, it is difficult to get away from shear stresses when fluids flow in narrow channels. It's great to hear you can deliver sensitive cells and maintain high viability. Can you please say a bit more about how you achieve this? Are you using shear fluids to reduce shear? Have you quantified total shear experienced by cells flowing in your channels?
I will love to answer these questions after the talk. The reason is my background is not in engineering, so I will take this question and get back to the person after okay, getting to our R&D team basically.
No problem. Well, that person has posed the question anonymously. So
okay. In that case, please contact us after the talk, and I'm happy to get our technical team involved into this.
No problem. That's good. So then the next question is, how large an area can be printed and how long does it take?
It depends on, again, this is really an open application area, right? It goes from what is the type of the cells, how well do they bind, right? In general, we do have experience of printing centimeters in that let's say that area, so centimeter type area. How long does it take? That's again it's hard to generalize. That's the problem, but we are happy to talk about very specific applications after the talk. I don't want to limit to one particular example and then leave the impression that is an absolute time frame for that. But in general, it is possible to go into centimeter areas.
Great. Yeah, and I mean the next question was related to that same question as well, so I'll skip that if you don't mind, Frank. The other one is great. Well, a comment about the technology. Great talk and great technology. So thanks for that. Are the printer nozzle? So let me just read it. So is the printer nozzle soft? I guess it is PDMS based, correct?
Yes, correct. The microfluidics print heads are PDMS, and that is a soft medical elastomer. Yes.
Cool. The next question is, again, thank you for the talk. Can you inject material directly into the center of the spheroids? Can you retrieve or pull out some cells from within a 3D structure?
So yes, it is possible to inject, to print on top of something else, or to place cells on top of something else. The first question comes to the positioning, right? So we cannot, it's not ideal to put something inside of let's say a gel matrix or matrigel or something like this, but it is possible to place it on top of that. And the same goes, I guess, for spheroids, even though that's the question of how big is the area where you want to place something on top. And regarding the second question, yes, technologically, it is possible to pull cells, to collect cells. Now, there are a couple of questions here. Number one is, how do you dissociate the cells? So if the cells are floating free and you come with the bioprinting head next to it, the cells will be pulled into the system. Now, the question really come about, okay, how do you dissociate the cells from that structure? And again, this goes back into the discussion, what is the structure? What can be the protocols to dissociate that? And the second part is also you know, pulling the cells in, they go in separate containers, but those microfluidics channels are quite long, you know, in the cell terms. So you know, pulling a couple of 10 or couple of 100 or couple of 10 cells, you know, up to 100 cells, you can expect to find some cells in the collection container, right? Lower than that, it's a question how much cells and where do you find that then in the chip.
Okay. A couple more questions. This one's from our friend Leandra in Brazil. So hello, Leandra. Hope you're keeping well. And basically, Leandra would like to know more about the velocity of the bioprinter, the speed with which it's printing, and the volume of the 3D constructs.
Yeah, I mean, you know, it's really the velocity and the volume. So it's a question again becomes what is the, and I'm sorry to, you know, have this generalist answer, but it's really, as you see from the capabilities, right? We can print a smaller scale constructs, or we can print a bigger scale constructs, right? And then the question is, if the time is how much time for, you know, a couple of thousand cells, or how much time for a centimeter wide construct. But I mean, it's not something to say that we can print a centimeter wide structure in you know, a minute or so. That is you know, that is not as you see from the concepts of the technology, that it's not something that is we can imagine, right?
Okay, good, great. So I think that's the main questions answered there. Thank you very much, Matija, for a very detailed and informative presentation. To everyone that's still listening, we do have a recorded version which will be available after the talk. And just wanted to end by thanking you all for your interest and your participation. And please do not hesitate to get in touch with us if you've got further questions. Thank you so much.
Thank you.